Welcome back. We've got a little bit more information about leaves on our plants. It's actually our last installment about plants. So if you weren't loving it, that's great. If you were, like myself, it's a little bit disappointing that we need to tidy up our lessons on leaves, but I'm sure they will come back to us at some point, although I don't think we're having exams. So you could be saved, but I don't know yet. So let's get started. I've called this the activity about leaves. It was originally going to be a microscope prac activity about observing a stomata, which we're going to get to in a second. But we're going to have to make do and use a photograph of the cells from the PowerPoint. I'll direct you to that slide in our class. So pretty much the activity here will help you answer page 3 of your booklet and hopefully give you a little bit more of a better understanding about the structure of the leaf. So our stomata, it's actually plural for stoma, so that's our singular if we're just talking about one particular cell. But really what they are tiny little pores on the leaf. And if you think about your skin has pores too. So look, leaves need to be able to be involved in gas exchange, so there needs to be a way in and out, and pores are the way for, I guess, this gas exchange to happen. So on page 3 of your booklet, you were asked to do a biological drawing of the lower epidermis of your leaf, so there were going to be samples involved. And the next part, you were asked to label the guard cells and the stoma. So again, we're going to make do. So I've drawn a biological drawing. I've kind of made it up, so it's probably the worst biological drawing ever. There's not really a title. I've used whiteboard markers, not pencil. There's no magnification, but we've got labels. So we're 1 out of 4, so it's not too bad. So very quickly, and hopefully you can still see me. If not, just keep on listening. We've got our guard cells, which surround each of the stoma, and they've got the capacity to open and close. So almost like a door, letting things in and out of the stoma. And if you look over here, I've written our stoma is responsible for taking in, I should have said taking, let me fix that while we can do on the spot edits. The joy of filming your videos in one hit. You just keep rolling, and that's a different coloured or tone of green pen. Anyway, our stoma is responsible for taking in CO2 and really struggling today, but it's important to fix your mistakes. Taking in CO2 and releasing oxygen. So that gas exchange essential for our input of photosynthesis, one of those things being carbon dioxide, and releasing our output, oxygen being that product of that photosynthesis equation. So that's, I guess, what those two components of the cell do. So if we look at our diagram, we've got our leaf cells, and again, we're looking at the lower epidermis. So I'll give you another example linked to skin. Your skin actually has 3 layers. So we sort of use the word epidermis for skin, but in this case we're using it for the layers of the plant leaf. So the lower would be the level or the layer at the very bottom. So very deep into the plant, we're looking at the bottom layer. So here you can see we've got our guard cells that surround our stoma, and they're almost like a little pore in the middle. So that's where the gas is moving in and out, and the guard cell can actually squeeze or release to open and close. So I've written down the bottom. This is pretty much the plant's way of breathing. So we often don't think about plants as living. They're pretty much breathing. They're definitely growing. They're doing stuff. So they're well and truly in the mix. So you're going to have a go at copying that diagram from the PowerPoint into page 3 of your booklet, making sure you label it, and you might want to add down these notes so that you've got them to reference back to. Cool. So I'll pop these pens down. let's come over here. This is my three-dimensional diagram of a leaf. You know, in school when you practice drawing like three-dimensional cubes, probably didn't go so well for me, and this kind of got worse, but maybe I do need a texter. Let me show you. So I tried to show you that this was like the top section of the leaf, and given that it's not flat or 2D, this is my sort of underneath bit. So please bear with me. So we've got the upper epidermis, which is the top, and then underneath our leaf, we've got the lower epidermis. So that's at the bottom or underneath the leaf. So the thing I want you to think about, and really this just sort of got worse and I tried to fix it, but yeah. I've drawn in our guard cells. So you can see here, lots of guard cells, the stomata in the middle. They're all scattered through the lower epidermis, so there's heaps and heaps of them. If we think about the upper epidermis, that top layer, there's not really many of them located. I think I've drawn like 4 or 5, but I really just want you to appreciate, not many up the top, heaps down the bottom. Lots and lots of guard cells. So there's 2 questions for you to think about on page 3 of your booklet. And that is, why are there more stomata underneath the leaf? Why do we have heaps here, not much up the top? And I'll give you a little hint. I might move this, I tried to section it off. It has a lot to do with the sun. So we know that we need exposure to sunlight to kickstart that photosynthesis process. I'm actually losing it, I'm cracking up over here. Anyway, so sunlight, we get those water molecules being split, photosynthesis gets started. But if we're thinking about gas exchange and being able to effectively utilize that water later on, it sort of makes sense that we've got our stomata located underneath. So it's actually cooler underneath the leaf. It's a little bit more protected. So it means that, yes, we capture the sunlight, but in terms of making use of water and effectively exchanging gases and finishing off that process of photosynthesis, it's much more effective for it to occur in that cooler conditions in that lower epidermis. So that's question number 1. Question number 2 that we need to think about is, if the guard cells close, so remember I said they've got the capacity to open and close, almost like a door, to control those exchanges of gas, why does the rate of photosynthesis decrease if they close? So I want you to think about the relationship between the inputs and the outputs of photosynthesis and how they're directly affected by the guard cells. So they're the things that I need you to have a really good think about and try and put pen to paper about to showcase your understanding of this concept. So a pretty short video from me tonight has not been my best for explaining things. So if you have any questions about this, maybe not about that bad diagram, I don't really think I can improve upon that, but anything at all, oh, I just tripped. Please don't hesitate to ask, and we're going to go through this during tomorrow's lesson. So please make sure you follow up if you've got any queries. Okay, I'm going to stop talking. Thank you.